All right, in this lesson, we're going to look at sort of the background for why it is cells have to divide. So we're going to look at cell size limits. And what I'd like to do is look at, first of all, the three factors that determine how big a cell can actually get, because there's actually a reason why cells are small. The first thing is this surface area, that's what SA is, to volume ratio. So surface area to volume ratio. Now remember, a cell's surface area is its membrane, which means the surface area is responsible for transport and the maintenance of homeostasis, right? So it makes sense that a cell is going to want to have a certain amount of surface area to carry out those vital life functions. As an example, let's look at what happens to a cube. Okay, it'll be easy to calculate that way. A cube, as we go from a cube that has a 1 centimeter side, a cube that has a 6 centimeter side, to a cube that has a 10 centimeter side. And that'll show you kind of what happens to that surface area to volume ratio. That'll be a model for what it's like with cells. So first of all, the area of a side of a cube would be the area of a square, right? Length times width. So for a 1 centimeter cube, this would be 1 centimeter squared, right? Now a cube has 6 sides, so its surface area should be 1 plus 1 plus 1 plus 1 plus 1, etc., until you get to 6 centimeters squared, okay? Now the volume is length times width times height. Well, 1 times 1 times 1 is 1 centimeter cubed. When you set up that ratio, what you see is you have a 6 to 1 ratio. At 6 centimeters, the area of a side is 36 centimeters squared. The surface area is 216, and I'm going to stop writing the units for now. That's centimeters squared. The volume is also 216, so you end up with this 1-to-1 ratio of surface area to volume. So when the cell is tiny, you have a huge surface area compared to its volume. But then it starts to grow, and as the cell reaches a certain critical point somewhere in the middle here, notice that the surface area and the volume are equal, and that's when it starts becoming dangerous for cells. So look what happens if we push this even further, though. At 10 centimeters, the area of a side is 100, right? The surface area then is 600. The volume is 1,000. Okay, and again, that's centimeters squared, and this one's centimeters cubed. In other words, you have a 6 to 10 ratio, otherwise known as a 3 to 5 ratio. Okay, so pretty quickly you start to see that the volume catches up and then surpasses the surface area if you get too big. When that happens, the cell starts putting too many demands on its DNA. You also start to see that the cell can't efficiently release waste, so waste starts building up. It can't bring in enough water to meet its needs. It can't bring in enough salts and sugars and so on. So again, it's kind of a bad way for the cell. So the surface area to volume ratio ideally should be large. In other words, large surface area, small volume. Two other things that are going to be determining factors in the cell size is, first of all, enzymes. Remember, even if I don't say it from here on out, everything inside of the cell is controlled by enzymes. Every step of a cell's life, enzymes are monitoring what chemicals are present, making new chemicals and so on, and also helping with the maintenance of the DNA of the cell. If you have too many enzymes, not enough enzymes, or there's some sort of mutation in the DNA, which is what produces your enzymes, you could produce enzymes that don't work right. That's going to mess with the cell size. You're going to have a cell that basically grows uncontrollably. The final thing is called contact inhibition. Now, contact, of course, means touching. To inhibit means to prevent something from happening. With contact inhibition, there's a picture down here. In a culture, we can demonstrate this. The cells will form a nice thin layer, a single layer in a culture. When the cells completely fill up the space, just like they do on your body, when they're touching other cells on all sides, they stop growing and reproducing. If you remove some cells, you interfere with that contact inhibition, and along the margins, the cells will start to go through mitosis. to divide and fill in the gap, okay? So contact inhibition will prevent cells from getting too large because once they're touching cells on other sides, they won't keep growing and dividing and so on. Now, when something happens and the cell gets out of whack and it continues to grow and divide when it's not supposed to, then you're going to have issues. So let's look at what happens. All right, so unregulated cells that are getting too large and that are growing and dividing rapidly, uncontrollably, are called cancers. So cancer, a tumor, happens when you start to have one little cell in a culture, just one little cell that for whatever reason starts growing and then dividing uncontrollably, just continues to divide. It ignores its contact inhibition signals, it ignores its surface area volume ratio signals, it ignores the enzymes, and it just keeps growing and growing and growing. That we call a tumor. There are lots of causes of cancer. There are the ones that are kind of the genetic causes, and then there are the ones that are the environmental causes, and we're going to look at those in class together. But an oncologist is a doctor that studies cancer and studies specifically the genes that are related to cancer. You may have heard a doctor say to a loved one who had cancer that their cancer metastasized. What that means is that it's spreading. So what these tumors will do is they'll grow and grow and grow, and then little tiny cancerous cells will break off, and they will enter the bloodstream, and they'll go to other organs. And the problem with these tumor cells now is, remember, there's cell communication. So the reason why cancers grow so rapidly is because once a cancerous cell gets started, it starts to signal surrounding cells to also grow and divide uncontrollably. So once cells from the tumor break off and go to other parts of the body, those cells in that part of the body will start to signal, and they'll start to corrupt the other cells in that part of the body, and you'll get a tumor there as well. So that's why cancer is such a nasty disease, because it spreads and then it corrupts cells in other parts of the body too. All right, last thought before we move on to actually looking at the real cell cycle is looking at how DNA in the cell looks. Throughout the cell's life, there are 3 different terms, and they all start with chrome, right, that refer to cellular DNA. So I want us to look at those terms right now. First of all, there's chromatin. And chromatin looks like this, and this is a really complicated diagram. I hope you can hold with me. There it is. Okay, that is a mass of tangled DNA that's all unraveled, all uncoiled. This is what DNA looks like through most of the cell's life. This is actually DNA that's open and usable, because what happens is enzymes will come along and say, oh, here's the piece of DNA that we need, and they'll pull it out and they'll use it and then they'll tuck it back in. That's the usable part of DNA, but the problem is, in preparation for cell division, this chromatin is going to copy. So now instead of one set of this tangled mass, you're going to end up with a copy tangled up in there as well. It's very difficult to separate the original piece of DNA from its copy when it's in this form. So in preparation for cell division, what cells will do is coil up their chromatin into structures called chromosomes. So here's what a chromosome kind of looks like. Okay, they just kind of coil it, coiled, right? And they do this, okay? These structures, these chromosomes, have 2 arms, and we'll get into the next unit on why there's 2 arms. But those 2 arms are called sister chromatids. So here's one sister chromatid, here's the other. What you need to know is that this sister chromatid is the original piece of DNA. This one is its copy, so that when cell division happens, this piece can be easily separated from this piece, and that's why they coil up into those structures. Excuse me, the last thing that you need to know is that this thing in the middle is called a centromere, and it kind of just holds the chromatids together when they're in that chromosome structure. That's spelled C-E-N-T-R-O-M-E-R-E. All right, so that's kind of an overview of what controls cell size, and also an overview of the different kinds of cellular DNA. You are ready to move on now to cell division.